polymerase chain reaction, or PCR, which is a method to increase or amplify the numbers of copies you have of a particular DNA sequence. This may be for various applications such as identifying viral infections or bacterial infections, or in forensic science where you're trying to increase the amount of DNA that has been discovered at a crime scene and you wish to DNA sequence the STRs, the short tandem repeats. But you can't do that, or it's very difficult to do that, unless you have the DNA in sufficient quantity. PCR is a method to artificially increase the amount of DNA that you have. This can be done very simply using a thermocycler machine, which you can see in the picture there. It's a three or 4,000 pound machine, which does this whole reaction for you in a matter of hours. Basically, if you've got one copy of a DNA molecule, it will take you through a cycle and you'll end up then with 2 copies. This is all automated. The next cycle you'll end up with 4. The next cycle you'll end up with 8. Then you get 16. Then you get 32, you get the idea. Followed by 64. Followed by 128. Now that's just a few cycles. You'll get 1 million copies in about 20 cycles. So you can see the potential of this method in rapidly amplifying the amount of DNA that scientists will have to use. Now, once the DNA sample is obtained, you are assuming that you would like to identify a particular sequence of interest. It could be, for example, an STR. Now, the DNA may well be in fragments. You may well use enzymes to cut up the DNA so far. Somewhere around the sequence of interest, such as an STR. What you then need to do is, within the machine, the DNA needs to be separated. It needs to be denatured, so it's no longer a double helix. This is done by increasing the temperature. Around about 96 degrees will denature the DNA. The next step would then be to copy the DNA. Now, in the cell, this is done using DNA polymerase, an enzyme, which will then insert complementary nucleotides. Now, DNA polymerase will not do this in the laboratory with single-stranded DNA, so it has to begin with something that is double-stranded. Now, to do that, we add something which is called a primer, which is a short sequence of DNA which should bind either side of your DNA sequence. So you have to have some knowledge of the kind of sequence either side of the sequence of interest, such as a short tandem repeat, so you can apply the right primer in this particular context. So the primers are added and they are stuck, or the technical word is annealed, to your DNA at around a temperature of about 68 degrees. This is still quite warm. It's enough to keep the DNA helix from reforming, but not too hot to prevent the primer from binding to its complementary sequence. Now you can see that we've got primers here opposite ends of the sequence of interest. So one primer is binding at the 3' end, and the other primer will bind at the 3' end of the opposite strand. And this is because the DNA polymerase will operate in one direction only, and it will catalyze the insertion of bases from a 5' to 3' direction. If you're unsure what I'm talking about with 5' to 3', we need to review the structure of DNA that you would have done at AS level biology. So the DNA polymerase needs to run from a 5' to 3' direction. Now we've got the primers added, and then we would add the DNA polymerase. Now the DNA polymerase will join the nucleotides to the DNA strands. This is done at about 72 degrees Celsius, which still keeps the double helix separated, but it happens to be the optimum temperature for this enzyme. Now, this enzyme was discovered in bacteria called Thermophilus aquaticus, which we find in hot springs. And this is preferable to use in the kind of DNA polymerase we'd find in our cells, simply because you'd have to keep dropping the temperature and raising the temperature again, and every time the temperature is raised to denature the DNA polymerase that you would have in your cells would denature, so you'd have to keep adding more. So this makes the automated process a lot faster by using this kind of polymerase that will not get denatured by the kind of temperatures that are used in the thermocycler machine. So what we need to do now is that we need to add nucleotides. Now, all the different types of nucleotides are added. This is not DNA sequencing where you try to terminate a chain. So all the nucleotides are added, and then what happens then is that DNA polymerase will run along the template, inserting the nucleotides at the right time. So as the DNA polymerase runs along the complementary strand, the nucleotides are inserted until it reaches the end of the DNA. Hopefully this won't be too long, but because you've probably got DNA in shorter fragments, this won't be a problem. It won't copy the whole genome, for example. So that will give you now 2 copies of the original DNA molecule, and then the cycle is repeated. Temperature is raised to denature the DNA. You need to add in the primers. Now the primers, don't forget, will bind at opposite ends of your DNA sequence of interest, binding at the 3' end of the original molecules, so that the new strand of DNA runs from the 5' to 3' direction. Then DNA polymerase joins, and at 72 degrees it catalyzes the insertion of the new nucleotide bases, making new complementary strands. So now in the next cycle we've resulted in having 4 DNA molecules. Go through it once more. DNA is denatured. Primers added. DNA polymerase catalyzes the insertion of new nucleotide to make a new complementary strand, and now we have 8 DNA molecules from the original.